Hi everyone. In this video, we're going to look at the processes of mitosis and cytokinesis in the division of cells. If you haven't watched it yet, I recommend pausing and checking out the video on the cell cycle and checkpoints before you go into this one. Mitosis and cytokinesis are the two components that make up M phase, which accounts for around 10% of the cell cycle. We're going to look at this in stages, but it's worth bearing in mind that this is a continuous process. The cell isn't stopping and starting throughout these steps. At the beginning of mitosis, during prophase, the activity of MPF will stimulate the breakdown of the nuclear membrane, and the chromosomes will begin to condense. As we progress into metaphase, we'll see the microtubule organizing centers, consisting of a pair of centrioles each, have moved to opposite poles of the cell, and the microtubules extending from them have attached to the kinetochores of the chromosomes. The jostling of the chromosomes from both sides will cause them to line up along the middle of the cell, along an imaginary structure called the metaphase plate. This is where that M checkpoint will come into play, ensuring that all of the chromosomes have been correctly attached to the microtubules. Assuming all of that happened correctly, the cell will progress into anaphase, characterized by the shortening of the microtubules and the subsequent pulling of the chromosomes to either end of the cell. Remember that once the sister chromatids have separated, they're full-fledged chromosomes. Again, this separation is due to the action of MPF, by causing the release of inhibition on the enzyme separase, which can effectively act as a pair of molecular scissors, cutting the cohesins joining the sister chromatids together. The cell will then progress into telophase, where it will build a new nuclear membrane around the sets of dorsal chromosomes and will begin the process of decondensing the chromosomes back to their typical G1 state. While this is happening, the process of cytokinesis will start to separate the rest of the cellular contents. Since this particular example is an animal cell, that begins with the formation of a cleavage furrow, generated by the contraction of a ring of microfilaments. This ring will continue to contract, eventually pinching the cell into two. The cytokinesis differs significantly between plant and animal cells, due to the need to build a new wall between the daughter cells in plants. So let's have a look at cytokinesis happening in a plant cell. First of all, vesicles containing materials for the new wall will deposit their contents along the center of the cell, roughly where the metaphase plate was. The contents of these vesicles will then fuse together to form a cell plate, and the cell plate will then grow into the new cell wall. Here we have a brief summary of the main steps of M phase. During prophase, the nuclear membrane breaks down and the chromosomes condense. In metaphase, the microtubules attach to the kinetochores of the chromosomes, causing them to align at the metaphase plate. The sister chromatids separate during anaphase, and the microtubules shorten to pull the now chromosomes towards opposite ends of the cell. In telophase, the nuclear membrane reforms and the chromosomes decondense. Cytokinesis, which usually starts either during late anaphase or during telophase, separates the cytoplasm and cellular contents, either by a cleavage furrow in animal cells or a cell plate in plant cells that will form the new cell wall. Thanks for watching, and if you haven't had a chance to check it out yet, please be sure to watch the video on the cell cycle and checkpoints.